Good morning, afternoon, evening. We are really pleased to have you join us for the Sharing Ocean Acidification Resources for Communicators and Educators webinar series. The SOARCE webinar is presented by NOAA's National Marine Sanctuaries and Ocean Acidification Program. Our goal is to provide ocean acidification communication tools to formal and informal educators and stakeholders across the country to promote a more integrated and effective ocean acidification education community. So for those of you just hopping on, there is a link shown here on the slide and a link in your questions box that you're welcome to follow to let us know where you're tuning in from today, that bit.ly. The title of today's presentation is CSI Oyster, a community science initiative on environment oyster interactions in Chesapeake Bay. I'm Jen Mintz, Education and Outreach Coordinator with NOAA's Ocean Acidification Program, and I'll be moderating today's session along with Laura Francis, Education Coordinator with the Channel Islands National Marine Sanctuary. Thank you all for being here today. We are really glad to have had over 140 registrants for today's webinar from all around the states and globe that are scientists and educators who are interested in ocean acidification science education. During the presentation, all of you, our participants, are in listen-only mode. You're welcome to type questions related to technical issues or questions for our presenters into the questions box shown here. It's in the bottom of the control panel on the right-hand side of your screen. Laura and I will be monitoring those incoming questions and will respond to them or pose them to our speaker at the end of their presentation. We always welcome your input as well on who you'd like to see as a future SOARCE webinar speaker, or what topics or resources you'd like us to cover in upcoming webinars. So if you have any ideas for us, please type those into the questions box as well, and we'll take those into consideration as we look ahead in the SOARCE series. We are recording today's session, and we'll share the recording with all registered participants via Google Drive and on the NOAA Ocean Acidification Program YouTube channel. So as we get started, we would like to understand or get a sense of not only where you're from, but your current role. So if you could go ahead and take a look at this poll question, go ahead and click on the role that best fits you. And if, of course, you don't fall into any of these categories, please go ahead and if you're in that other category, just go ahead and type your role into the questions box as well. So there's a great variety of folks. We'll keep this open for just a few more moments. It looks like we have almost equal representation, educators and scientists. Okay. Great. So as you all can see, we've got a great mix here. Educators, some science communicators, and researchers on the line. For those that are in the other category, there are resource managers, permitting consultants, a nice variety of folks joining us today. Thank you for sharing that. So we are really pleased to have both Emily Rivest and Bart Merrick speaking with us today. Dr. Emily Rivest is an assistant professor at the Virginia Institute of Marine Science, William and Mary, and her research focuses on understanding how ecologically and economically important species like oysters, hard clams, and American lobster will respond to climate change. She is an expert in ocean acidification research, conducting experiments in the laboratory to simulate future water conditions, and using oceanographic instruments to characterize the dynamic coastal environment of her study species. Emily is passionate about doing science that matters, often collaborating directly with members of the aquaculture industry. She is also an avid science communicator, aiming to share the importance of her work with policy, industry, and public audiences, as you'll see today. Bart Merrick has been an environmental educator for 25 years, working with teachers and students from Massachusetts to Virginia, promoting environmental literacy and fostering understanding, awareness, and respect for the natural world. Upon receiving a master's in environmental science, Bart worked with the Maryland Department of Natural Resources as the education coordinator for the Chesapeake Bay National Estuarine Research Reserve. And currently, Bart is the education coordinator for the NOAA Chesapeake Bay office's Environmental Science Training Center, focusing on integrating the use of environmental science and technology with field-based learning experiences. So I think without any further ado, Bart, pardon me, I will go ahead and hand it over to you.
Wonderful. Thank you very much, Jen, and we appreciate it. And I'll go live for a second just to say, oops, that was a quick second. say hello to folks. And so hopefully you all are seeing what I think you're seeing, which is just a quick screenshot of that ask that we put out there in terms of as you arrive. And we just, and I thank you all for chiming in on this already. But I find like in these webinars and these conversations that we're about to have, seeing where people are from and, you know, kind of having a way to kind of poke around a little bit and get that geographic place-based opportunity to see, you know, where our colleagues are working and where they're from is kind of a nice opportunity. So I'll just call that Laura did put a link in the chat to this Padlet, which is a map-based Padlet. And if you haven't had a chance yet and you want to, we'd love for you to kind of add your place there as well as your name and maybe your connection to the education ocean acidification world. So let me go full screen here. I think I'm not quite there. And so Emily and myself are both here to share with you about a project that we have collaborated on that you heard the name of it already, CSI Oyster, the Community Science Initiative on environment oyster interactions in the Chesapeake Bay. So just in terms of introductions, maybe Emily, I'll hand it off to you first, just to introduce yourself real quick verbally, in addition to what Jen had to say, and we'll show you where Emily is from at the same time. Oh, Emily, you might be on mute. Sorry.
Hi, everyone. I'm Emily Rivest. Thanks for coming today. We're really excited to talk about this project we've collaborated on and share it with you. I'm based at VIMS, as Bart has highlighted, although I'm not physically there this afternoon. But I have been a professor at VIMS for 5 years, and my program focuses largely on ocean acidification and its co-stressors, mostly in the Chesapeake Bay region. And Jen gave a lovely introduction, so I think I'll leave it there.
Cool, thank you, Emily. Yeah, and then I, you know, also as Emily and Jen kind of pointed out, well, Bart Merrick, I work in the NOAA Chesapeake Bay office, and one of my primary responsibilities that brings me to this webinar is really to support kind of environmental literacy education connected to NOAA science, and our partner, Chesapeake Bay Region Science, connect that science to the education community. And yeah, and like Emily, I also am working from home, like many of you probably these days too. And I'll just remark that like it's really exciting to see people coming from, you know, across this country to share in this conversation about you know, about ocean acidification and the education aspects of some of the work you all have done, and providing us with an opportunity to share a little bit about the work that we've done in the Chesapeake. So what are we up to? So in this session, there's a couple things that we hope to dig in a little bit. A little bit is context. So a little bit about coastal acidification and kind of just a little content on that. And then a little bit about the environmental literacy, environmental education context that we work in the Chesapeake Bay region. So I think that'll hopefully be informative. And then also about the notion of using and kind of exploring community science as an opportunity to both conduct some of the science that needs to happen, but also engage communities in learning about these resources. and then we'll dig into the project. And we'll kind of tell you a little bit about some of the education components of it, and then the details about the project and the results. And at the end, we'll wrap it up with some successes and some challenges that we have met along the way, and then just open it up for discussion right at the end. One thing I did want to note is that we did want to kind of support like an open chat type feature here, and we will be asking throughout this presentation for a response or two within the chat. But again, we'll keep it open. And so both Emily and I have another window open, which is the link that you can see on your screen. This one that's another bit.ly source chat. And that space, I would say, is exactly what you're using it for, which is great. So you can add some comments here, tell us who you are, tell us where you're coming from. And then later on in the presentation, like I said, we're going to do a little chat exchange. And we just hope you have that open. You can play with that there. So that's it for the intro piece. So I'm going to hand it off to Emily to kind of kick off the actual presentation part. And I think you can just steal that. Yeah.
Wonderful. Thanks, Bart. Well, first, I want to start by acknowledging all of our partners and funding sources and other contributors to this project. A community science project involves a community of people, and we're very lucky to work with such amazing people and funding partners. So I want to thank NOAA's Ocean Acidification Program for funding this work, kind of through an internal opportunity for projects led by NOAA employees, and that allowed me to connect with Bart and start this cool collaboration. So it's been wonderful. As you'll see through this project, we partnered with 2 high school teachers, one in Virginia and one in Maryland, who have been integral to the success of this project and the development of the ideas coming out of it. And then a plethora of students, graduate students, undergraduate students, volunteers, and Elise, who works with Bart, have all helped keep this project going in various ways through mentoring and time on the water, etc. So,
Emily, one quick thing. Something might be a little wonky. I don't think we see the same thing you see in terms of the slide.
Sorry. What can you see?
So Emily, I'll just give it, yeah, there we go.
Yep.
Can you see it now?
Yep.
Okay, great.
Sorry, thanks.
Thank you for interrupting me and letting me know that. Okay. All right. So I wanted to start off broad with this talk to show you where I come from in terms of thinking about ocean acidification and coastal systems. And for the research that I do, I'm a physiological ecologist, and I study through that the interaction between organisms and their environment. And one of the things that we're really interested in thinking about with ocean acidification, we have this long-term environmental change that's happening, but it's happening across different temporal and spatial gradients of environmental variability, which I'll talk about more in a minute. And so a lot of the challenges that we face in the scientific community in anticipating what biological consequences will be is in part being able to anticipate what habitat conditions will look like in the future. What will oysters experience at different places in Chesapeake Bay in different seasons? And we know this natural environmental variability exists. It has existed through evolutionary time. And so the physiology of the oysters, their ability to tolerate stress, to tolerate change in their environment, might be affected by the local conditions that they experience. So that interaction is really important. And then closing the loop here to tie it all together, you know, what aspects of their physiology are important for allowing them to tolerate this human-caused global change that Earth is experiencing right now. So this is a very sort of broad framework of questions that guides my scientific research. And from here, we'll sort of zoom into how community science plays a role in this, because it plays a very important role. And it's something that I've always wanted to incorporate throughout my career. And finally at VIMS, I was able to do that through this project, which is wonderful. So if you are a repeat attendee of these webinars, I'm sure you're familiar with ocean acidification, but to make sure that we're all on the same page, just wanted to talk about a few relevant details that are important. So ocean acidification as a global process is caused by humans putting too much CO2 into the atmosphere. That the ocean serves a very important role for our climate, and that it absorbs a bunch of. And in the water, that CO2 undergoes a series of reactions that result in two chemical symptoms of ocean acidification that are harmful to marine organisms. We have an increase in hydrogen ions or a decrease in pH that disturbs internal cellular environments in organisms and disturbs biochemical reactions that help them survive and grow. And we also have a change in the availability of carbonate ions, which is an important building block of calcareous or calcium carbonate skeletons and shells. And so we know these mechanistic relationships and how changes in water chemistry affect the biology of a lot of the animals that live in the ocean. And we've gotten at that through laboratory experiments in facilities similar to what I have at VIMS, where we can change the chemistry of the water in a tank. We can put our animals in there and we can measure what happens to them. But of course, doing experiments in the laboratory, you are assuming that what you find is what will happen in the natural environment when the ocean becomes that acidic, for example. And so one really important part of the scientific process is being able to translate the results from the lab into the field. And that is where community science can play a really important role, because in the real world, there's all this natural environmental variability that we never have enough space, time, resources to simulate in the laboratory. And so getting involved in community science can help with this process of extending our lab discoveries into the field, in addition to having lots of other benefits, which Bart will talk about more. And boy, do we have a lot of natural environmental variability in Chesapeake Bay. Ocean acidification as a global process is occurring across these existing temporal and spatial gradients of chemistry. So for example, Margie Friedrichs at VIMS produces these forecasts of bottom water pH, and the purple triangle here is where VIMS is, and Bart is located up here in Maryland. And you can see that on March 3rd, that the bottom water pH was not consistent throughout the entire bay, and oysters live across this range of much of this range of pH. Here is a time series of pH collected on the pier at VIMS in June. And you can see how dynamic from day to night and day to day the pH is. These lines I've put on here, the pink line is present day average open ocean pH, and the red line is the 0.3 pH change we expect to see in average ocean pH by the end of the century in the open ocean. And so you can see right now in Chesapeake Bay, these oysters are on a really wild roller coaster pH throughout their lifetime. So this degree of spatial variability in pH is of the same magnitude that oysters can experience over 24 hours. So a community science project is really exciting because we get to work across this wide range of conditions. And of course, in the estuary, we're not only thinking about coastal ocean acidification, we also have to consider coastal acidification, which will compound the degree of chemistry or acidity that the oysters are experiencing. And the way we define coastal acidification is that in addition to the driver of atmospheric CO2 causing acidification, we have other drivers of acidification like freshwater inputs from the tributaries, runoff from the land. We have nutrient inputs that cause eutrophication, stimulating phytoplankton blooms, and as the dead phytoplankton are being decomposed, we have changes in CO2 and pH analogous to ocean acidification that happen from that. So studying ocean acidification in an estuary in the coastal zone is quite complex. And in addition to that, this natural variability is further accentuated by the different buffering capacity of river water. And this is a newer result published recently with co-authors at VIMS. And they looked at the alkalinity of, or the buffering capacity of these different tributaries of Chesapeake Bay. And you can see that plotted here. Each different colored line is a different tributary. And so you can see that the alkalinity varies through time, and it's also different between the tributaries. And when we think about buffering capacity, it's the how much the water can resist changing in pH. So if you have a high buffering capacity, it can resist that pretty well and it will be more stable. If it's a weak buffering capacity, then the pH could change much more quickly with a small input of CO2. So the susceptibility for intense acidification is dependent on this buffering capacity. So depending on which tributary the oysters are in, you know, these oysters in the Potomac with a much higher alkalinity are going to experience less dramatic change in chemistry than oysters in tributaries with very low buffering capacity. The other thing to think about is that as climate change progresses, we are going to see wetter, more rain in the Chesapeake Bay region in particular seasons. And more rain means more stream flow, and that means lower buffering capacity. So there will be certain times of year, for example, when oysters will be more likely to experience more intense coastal acidification. And if that weren't enough, in this complex scenario for studying ocean acidification and environment oyster interactions, we have other stressors in estuaries too, of course. And so in addition to gradients in chemistry, we have gradients in salinity, we have seasonal hypoxia, we have all this constellation of stressors that can blink on and off, be more intense in certain places or in certain times, to think about what all could be impacting oyster health. So when thinking about how to link natural environmental variability with oyster performance in the context of ocean acidification, we say, okay, if we want to study this through time, we need to know how these things are related. What resources do we currently have? Well, there are a lot of groups in Chesapeake Bay that are measuring water quality. We have the NOAA program, we have the NOAA Chesapeake Bay interpretive buoys, we have a program that collects water quality data. And we have a couple of programs that routinely collect oyster data, and these are, I should say, Virginia biased, so there are more in Maryland, I'm sure. But one of the things I noticed when I started at VIMS is that there weren't programs that were collecting these two things in concert in a co-located way. And so it's really hard to draw relationships between them, to think about how all these things are related in this very complex, dynamic coastal environment without that. In addition, we have this opportunity to insert environmental literacy into this Venn diagram. And by generating this program based in community science, we have the opportunity to enhance environmental literacy in our region surrounding the connection between water quality and oysters in the context of ocean acidification.
Sorry,
Yeah,
go ahead.
go ahead. It's okay.
Oh, yeah. So I like just thinking about one of the other contexts. So Emily just laid out the variability that the oysters have to thrive in as part of the context. Part of the other context here is that we have, you know, in the Chesapeake Bay region, we have a lot of, you know, it's the world's large, or the country's largest estuary, right? And it's this incredibly vibrant body of water, and yet it's challenged. And one of for addressing that challenge is to promote environmental literacy. So we can click to that next slide. And that's a big part of what this project is about, like kind of like find that sweet spot in the Venn diagram where students, research professionals like Emily, educators like myself, and the teachers that we work with really get to dig in, support the science, but also develop a greater understanding of the dynamic Chesapeake Bay as a system and the process of doing science like this. We can click that next one. And so we had two kind of primary objectives here, like, and one of them, you know, was to advance environmental literacy and public awareness about ocean acidification, and then develop that community science approach to collecting data on the interactions between, you know, the water quality, the oysters, and the chemistry of the Chesapeake in these two areas that we'll be talking about. We can click. Thank you. And so the CSI, like named after the crime scene investigator thing, the CSI Oyster project, right, is all about doing that and engaging students in part of this to look at different techniques for measuring water quality and oyster size and exploring that relationship between that environment that they're in and the oysters themselves. Click. Thank you. And one of the things that really helped us here in terms of when we're approaching some of the educators that we did work with and their administrators was the fact that in the Chesapeake Bay region, we have what's called the Chesapeake Bay Watershed Agreement, which drives a lot of our work to both understand the change in the system, how we're approaching the conservation of that system, and how we're supporting the community around it and understanding what's going on. And you can click to the next slide, Emily. And one of the things I feel personally very fortunate about is that in that Chesapeake Bay Watershed Agreement, we actually have one of these goals that the entire watershed states are working towards is environmental literacy, which gives us a lot of, I'll call it latitude, but I think really just, you know, entree into conversations with partners about not just the science, not just how the Chesapeake Bay is doing, but how do we engage the community and students specifically in this. And one of the goal specific, well, a specific part of the goal for students is that these students will actually participate in environmental literacy programming. And the name of that environmental literacy program is actually Meaningful Watershed Education Experiences. So we'll click to that next slide. And I'm curious, and it's kind of hard to tell on here, but just maybe think for yourself a second, or if you want to put it into that webinar chat. Has anybody here ever heard of the Meaningful Watershed Education Experience? Just pause for a second there, just think. And depending on where you are in that geographically dispersed Padlet that you shared with us, you know, if you're not from the Chesapeake Bay region, you might not have heard of it. In NOAA, we have a Bay Watershed Education and Training grant program, and we focus on the MWE there. So if you're maybe at a Natural Estuarine Research Reserve, which a couple colleagues are from that, or potential like maybe sea grant partners that are out there, or other yeah, sanctuaries, marine sanctuaries, you might have heard of some of these things. So good, I'm seeing some BWitness, I'm seeing some yeses, this is awesome. So we can click to the next. But I think just to provide the background, this whole entire program, this CSI Oyster project from the education side was framed around the Meaningful Watershed Education Experience. And so I want to just spend a second on what that is. And the MWE is that project-based approach to K-12 environmental literacy programming. And the whole deal is you take locally relevant environmental issues and you explore them with the students in you know, in the goal of connecting it to standards-based learning, but also ensuring that the students are seeing that relevancy, having the opportunity to engage in that issue, and ultimately taking some action to try and address that issue as well. So we can click to the next one. So the idea is that MIWIs are really a powerful tool in the education environment for supporting basically 21st century learning skills, right? So they help us with that. They help us engage students because that local relevancy helps give people a reason to learn about some of these more complex topics like ocean acidification or estuarine acidification for a reason, because there's something that they know about and are familiar with right in that community. And then ultimately, after we work through that sort of stuff, then it's an opportunity to kind of get to the point where we're actually doing something about it. So we'll go ahead and click. The MIWI itself has 4 components to it that are really important, and we call these the essential elements. And the essential elements of the MIWI are actually what the students do. So this is the students are actively engaged in these things. So we can go ahead and click to the next one, sorry. And the first is issue definition. And that's where, like with this project, ocean acidification or estuarine acidification is fundamentally the issue, or maybe even a more basic way to look at it is like how the environment impacts the things that live there, the oysters specifically. And that issue is something that students can focus on, develop their own questions about, see where the problems lie, and connect to that through background research. We can click again. And then it's also important that as part of this MIWI that students have that opportunity to go outside and do something about it. And that's one of the best things I think I've found with this program is that students were actively engaged in going to a neighborhood spot where they have been 100 times, and looking at the oysters that were there, actually talking to some of the community members that worked or kind of fished from that spot, but engaging in that outdoor field experience to investigate that issue in a meaningful way. We'll go to the next one. And then the idea is whatever data they're collecting, whatever their work they're doing to look at this issue, they bring that back to the classroom, or they bring that back wherever, and they actually do some work with it to understand what the data might be telling them, to be able to communicate about what that data is telling them, ultimately so they can make a claim about what the conditions are out there, like how is water quality impacting the oysters in this community. And then the final part of it is often where students have the opportunity to say, all right, we have a challenge here. The water quality here, or the conditions in this this harbor where we're putting our oysters are not really good for the oysters. We need to do something about that. So what do? They can, all kinds of stewardship and civic action opportunities that could include communicating to the public that might be there, putting in ways of managing runoff that are part of the issue in this community, or even personal actions that they might be able to do, like just simply keeping their dog waste from ending up in that creek. So the stewardship action is key, and it locks it all down into hopefully supporting what we hope is environmental literacy throughout the community. So you can go ahead and click. And we, we did anchor all this work and use the strategy of community science. So actual science conducted by students that was relevant to Emily's work on ocean acidification and its interactions with oysters. And that community science turned out to be a really powerful tool for enhancing both the scientific research and monitoring, building capacity to measure things that are geographically disparate. It also provides that opportunity for the students to have that hands-on learning in very real ways, and then it can help address some societal challenges too, depending on what the citizen science project looks like. And that's kind of a neat and powerful tool that we really thought a lot about engaging throughout this project. So we'll go to the next slide. A little bit about this project and kind of just what it all looked like. I think I'm still going here. Okay, cool. So in terms of involvement in this project, we had around 100 high school students. We had a number of graduate students working with Emily. Emily herself, right? A couple education outreach folks, myself and some colleagues at the NOAA Chesapeake Bay office. And then summer students, both interns that we had and interns that Emily had to support this work. Because even though the students might go home for the summer and they might not be in class, the oysters are still in the water. So it's important to keep that continuity going as best we can. And so what do we do to support the education aspects of this program? So we had 4 components: teacher professional development. We developed a really rich monitoring manual. We did some in-class programming, and then we also conducted evaluation related to the work. So we can go to the next. So in terms of the teacher PD, that looked like one-on-one training with the educators in Virginia and Maryland. And then we were out there kind of right next to them during those first initial sampling opportunities with students, with each other, to make sure we had everything nice and set. And then there was ongoing support. So all you needed to do was call one of us and we would be there to help you with some of this work. And then the monitoring manual, like that's, we all know instructions are pretty important. If you've ever put together IKEA furniture, I'm sure you diligently read and follow every instruction there. And so we applied that in this project too, and actually assigned students roles when they're out conducting these field investigations. And then those roles came with step-by-step guidance about how to do the practice, how to record the data. We can go to the next. And then in class, to ensure that we did make that connection as a local issue to some of the bigger challenges or some of the drivers of the issues related to coastal and ocean acidification. We did some in-class programming where we met directly with the students. We provided presentations on issues in the Chesapeake and how they related to ocean acidification, and then actual activities and lessons. Like you see the young ladies in this picture doing kind of the classic ocean acidification activity by putting, exhaling into some water with the pH indicator on there and changing the pH just with their CO2 they're exhaling. We go next. And then lastly, we did work to try and understand both the initial conditions that the students were experiencing in terms of did they know what ocean acidification was, and also thinking about how ocean acidification might impact oysters. And these students actually started off at a really high bar. So we didn't actually see a whole lot of change in the pre and post, but I think that's a credit to the educators and the teachers we worked with, especially the teachers in Virginia. So those were the wraparounds that kind of drove the education programming part of it. We're just putting the objectives back up here as we transition to Emily talking more about the technical aspects of this project and some other results.
Great, thanks, Bart. So to achieve these environmental literacy goals and also to try to get at that knowledge gap I mentioned earlier about providing a way for community members to really help advance ocean acidification science, we developed this community science data collection program called CSI Oyster in partnership with our teacher partners. And so each school established several sites. You can see here the more widely spaced the sites of the Chesapeake Bay Governor's School and a couple sites up here for St. Michael's High School, and we had one site at BIMS that we maintain. And the teachers helped us select the sites based on their a priori hypotheses about sites that would have different levels of water quality, good, medium, bad, if you will. We provided them with oysters, all from the same batch that they deployed at their sites, and they, with their students, would visit the sites every 2 to 4 weeks and measure water quality, including the chemistry of the water, and survival and size of the oysters. And the way that we set up the protocols for this data collection program would allow us to compare different techniques for measuring water quality and for measuring oyster size, so that we can inform the design moving forward of this data collection program to collect good quality data about water quality and ocean acidification. And then this data set could also be used to explore that relationship between environment and organism through time and also among these different sites. So for the comparison of water quality measurements, each teacher was provided with a handheld YSI instrument that allowed them to measure temperature, salinity, dissolved oxygen, and pH. There was also a little hobo logger on the crate that would collect a higher frequency time series of salinity and temperature. And then we trained them using we trained them to use the international best practices to collect and preserve water samples, which would be transported back to VIMS, where our lab would measure pH and total alkalinity using the methods that we typically use. And this was a data sheet that we had where they would collect some of this information just to give you a flavor of what the students were doing. So, they could confirm that they completed various aspects of the project. They would also, after they did the water quality part of it, they would pull up the crate of oysters and make note of how many were alive and dead. And also just general observations about what else was on the crate. And I've had other students choose to use the various different bits of these data in other ways. So it's really lived much beyond this project. For measuring oyster size, we measured shell height in a couple of different ways. One was using calipers. So we trained the students how to use calipers. And the other was through an invention of mine, which is a 5-gallon bucket that anyone can find and carve out to make a photo bucket. So with a lot of the point of these methods comparison is also to evaluate, whether these more accessible methods for measuring these different aspects of the environment or the oysters provide equally good quality data as the more expensive or traditional methods that we would use in our research. So anyone could place their phone or tablet or camera here and take a standardized image of each oyster in their bag with the identifier name there in dry erase pen and a ruler for calibrating the image afterwards. And so here's what the data sheet looked like for that. Each oyster got a unique identifier where they could write the shell height here and confirm the picture was taken and make any notes about it. For example, if the oyster was a double with 2 shells fused together, that's really useful for the person who's analyzing the picture. So as I mentioned, each school had a core site that they visited every 2 weeks, and then a couple of other sites that they visited at a lower frequency. So some of the results that came out of this, we compared the photo analysis and the caliper measurements for the oysters and found they were in great agreement. So teachers can, in the future, could choose which kind they would want, and the data would be we would be able to integrate that data set that came from different methods, which is really nice. And I'm gonna see if this is going to work. No, I don't think it's going to work. Well, let's move on to oyster survival. So here are some of the data for oyster survival. We have our 7 sites here, 6 sites here, and the different colored lines. And starting out at 100% survival, you can see through time which sites had better survival or worse survival. We can see there's a big difference. So this is one where we would like you to use your chat and help us think of some reasons for the pretty extreme difference in survival between Maryland and Virginia sites. So these green and blue lines here are the 2 Maryland sites. And you can see that there was a lot more mortality at those sites than in our 4 Virginia sites. So what aspects of water quality or other reasons might we see lower survival at these Maryland sites? And Bart, I can't see the chat, so if you can help provide some reflection on the responses, that would be great.
Yeah, absolutely will. And if folks want, they can actually just grab their phones and take a picture of that little QR code, and I'll bring you right to that Padlet chat as well. So we have poorer DO in Maryland. Sad. It's a good one. And but maybe less buffering from freshwater input. So lower DO, maybe less seagrasses, or just buffering habitat. Temperature is another one that's coming in. More thumbs up, kind of giving some thumbs up for DO and the buffer difference, like the different buffering capacities in the areas. I like it.
Great. So to give you some of what we found, our St. Michael's Harbor site actually, DO wasn't as informative in our data set in this particular year as some of the other variables. At this site had the lowest average pH, although not necessarily the lowest minimum pH in the data set. And combined with low temperature and low salinity, I think really impacted the oyster survival at that site. And yeah, at least within this data set. Whereas in Virginia, this Vims Pier site had much more favorable carbonate chemistry combined with a higher salinity. So we had, it was interesting, one of our Virginia sites, this Piankatank River site, this gray line, is a site that had equally low salinity as our two Maryland sites, but and often lower pH as well, but it had other characteristics that made it different. So there's something unique about these Maryland sites. I think it's this combination of chemistry, temperature, and salinity, and potentially other things as well that impacted oyster survival. So we also measured oyster growth, and so this is average shell height through time. Again, with the light green and light blue time series being our Maryland sites, and the other four being our Virginia sites. And the oysters in Virginia grew faster, with this purple line, Merroir, being in the lead. And when we look at the water quality data, some of the correlations that we see are that the Maryland sites have low saturation states. And so this is where that combination of temperature, salinity, and pH that's what this is resulting in. These very low saturation states, which is an aspect of chemistry that is directly relevant to calcification, or which is what we're measuring the growth of the shell by measuring the longest shell length. If we just look at the Virginia sites, we see that the saturation state at Piankatank is the lowest. We have the lowest pH and salinity in Virginia. It's higher than in these Maryland sites, but that's explaining why growth is slower in the Piankatank River. But with Merroir, beyond that, there wasn't anything that stood out among the data that we measured to give us a hint of why growth was so good at the Merroir site. And so we, of course, one of the limitations of the program that we set up here was that we weren't measuring chlorophyll A or sort of dissolved organic material in the water that is accessible to particulate organic material in the water that the oysters might feed on. So that's definitely something that would be great to add in the future, and may explain why the oysters did so well in Merroir. So when thinking about outcomes of this project, we were able to observe how oysters respond to seasonal changes in their habitats by collecting these measurements at regular intervals through time. The students and us gained a better understanding of Virginia oyster habitats, how they differ among these different places and throughout the year. The students could identify different drivers of oyster growth at each site by comparing the different water quality variables, and could also use the data set to identify sites with the best water quality for oyster growth. And I will echo what Bart said, that one of the observations that I had in working with the students in this project, and one of the biggest impacts that they would talk about, is just the experience of going out to these sites regularly throughout the year and seeing the oysters grow themselves. And they got so invested in the oysters that they were measuring, and you know, why oysters at one site would be doing better than others. So it was just a wonderful immersive experience for them. And so in terms of outcomes of this project, we have the results of these comparisons that we can put into action for future use, and resources like the student monitoring plan that you all are welcome to ask for. We'll happily share those with you. And also, one of the things that we highlighted was this, is that you do really need to have these paired samples that, both of these types of water quality measurements, particularly for the carbonate chemistry, are necessary in this very dynamic coastal environment. And I think, Bart, is this me as well?
Sure, and we can tag team it, or I can
sure. Yeah.
Color comments.
Yeah. So we engaged on this really cool collaboration with teachers that that were both new to us and that we'd worked with in different ways before. For some of the challenges with this project in terms of education is that not all students that the teachers worked with had access to this experience, or they had access in different ways. So depending on how the teachers organized their classes, or how they decided to incorporate this in their teaching at their school. For one teacher, it started off as being a whole class activity and transitioned to a smaller group of students that regularly helped out, while another teacher would cycle through students and take small groups. But there were certain students who would just beg and beg to go each time. So it was a challenge to make it accessible to everyone. It was also a challenge to connect students from both schools. I think that would be a neat addition in a future iteration of this work, to think about how to get the students interacting between classrooms. We weren't able to involve the students in data analysis as much as we had anticipated. Part of that was that we had envisioned maybe if there were certain students that were really interested in doing more with the project, that they would be involved in the photo analysis or working with the data sets that everyone generated. And there were some who initially expressed interest and didn't quite follow through. We involved undergraduate and graduate students in that way more successfully. So it would be great to develop that aspect of the project in the future as well. And then we had in Maryland identified a teacher partner at the time we wrote the proposal, but by the end she had been promoted within her school and was no longer a teacher. And so we had some challenge with changing staff, which happens. Research related, it is a big time commitment for everyone involved with these investigations. And I think one of the lessons we learned that was really important was that that flexibility with working with the high school teachers is critical. And from a scientific research perspective, you want to be, have consistency and control and a standardized process. But in order to make this work for the teachers as they're realizing what it's like to collect the data as we go, flexibility was really key.
So Emily, just kind of like a time check, and there's a number of questions in the chat and out there too. And we have like 4 minutes, so maybe just the next slide we can put it up, or I mean people, yeah, or and offer some question space just while we have it.
Yeah. Yes, I would be happy to. So I'll put the successful aspects. You've already seen the success in terms of what we talked about with enhancing environmental literacy of the students and the data we collected and the partnerships we developed, which will last beyond the time frame of this grant. So yeah, I'd love to open it up for questions, and thank you all for learning with us today.
Thank you.
Thank you both. I think it was just so valuable to hear both sort of of course, the education, environmental literacy piece and how that was set up, and then actually seeing the methodology and some of the results. So we do have a variety of questions, and I know some of them were asked in the Padlet chat and some were answered in the chat. There are a few that weren't. So we'll just open it up. I think there was just one, I know this project took place before COVID, but there is just a question. I know, Bart, you opened it up about MeWes, and someone was wondering, you know, was MeWe modified during COVID, virtual education? How is it financially supported, and what is the level of participation? Schools, students, and staff, just to give folks a sense of that.
Yeah, I mean, that's a big question, and the short answer for that is yes, MeWes have been modified over the past year due to COVID, and the challenges related to getting outside with groups of students just didn't do it, right? But a lot of our partners that are working on developing MeWes were able to get their students outside and maybe in their yards or somewhere close, and really come up with some innovative ways of still connecting to the issues, conducting investigations, and taking action. A lot of that action was, you know, about letter writing, PSAs, poster and artwork things. So it sort of transitioned from a lot of on the ground stuff to more appropriate stuff in the virtual environment. And as far as the funding piece goes, you know, from my perspective, we're continuing to fund MeWes at the same level that we've been able to, and in addition, we're recognizing the challenges related to getting students outside. So we've actually kind of convened a number of conversations and encourage our grantees and partners to think about, you know, how to pivot during COVID and how to emerge from it strong, you know, as well as they can to move things forward. It's challenging though, because a lot of organizations had a really hard time during this period, and yeah, and continue to struggle. So hopefully that answers it.
Yeah, I've done it.
I'll add to that that we've continued with the data collection part of this with our Virginia teacher this year, and it has, although COVID has impacted all of us in a lot of ways, it has been one way that she has been able to interact with her students safely. You know, they don't see each other in the classroom, but they can meet at the site and do this together. And it's, you know, socially distanced and masked and everything. So that was a nice part of it.
That's great to hear. So I just want to be mindful of time. So I'll just ask one last question, and this is sort of, you know, Emily, you're continuing to work on this project. One of our participants is wondering if you could see this program expanding to manipulative experimental tests with some of the hypotheses that were generated here.
Yes, I could certainly do that, and we've leveraged this in the year that the date that this particular project was done. I had an REU student who took the oysters that came from this site and tested the effect of environmental history on stress tolerance. So we used the citizen science, the community science data set, which characterized the environmental history of these animals for the past year. We brought all the animals back to VIMS, and then we put them through the ringer and did some stress tests. So it's a really neat framework for doing a lot of different things, and certainly you could use these as preliminary data to design field, transplant experiments, lots of different kinds of experiments.
Great, that's wonderful to hear. So I think we'll have to wrap it up there. I do want to just offer folks, I know in the Padlet, Bart offered his email address. We'll put those both in the chat. If there are follow-up questions for either of our presenters, please go ahead and let those, let those happen. Let the conversation continue, either in Padlet or otherwise. And thank you, Emily and Bart, for a really interesting and engaging presentation. As you'll see in the Padlet, I think that, people learned a lot, were really engaged, and just thank and give you both kudos for today's talk. So we will go ahead and share the recordings and links to any of the resources that Bart and Emily mentioned, and in the SOARCE webinar archive on the Google Drive, and that will be early next week. And as I mentioned, any questions, if you'd like to continue the conversation, please feel free to email Emily or Bart directly. And yeah, we just want to thank you for attending the SOARCE webinar today. We always welcome feedback, further questions, suggestions for topics, and you will receive, as we mentioned, a follow-up email, so feel free to offer us input there. And you will have the opportunity to provide feedback with a survey that will appear right after we hop off today. And we really, really value that and appreciate if you could just take a few moments to complete it. That feedback is really what allows us to continue hosting this series and to inform NOAA of its impact. Yeah, so we'll share that recording of the webinar shortly. It will make its way onto the NOAA Ocean Acidification Program's YouTube channel, and a PDF of the presentation will also be shared as well. And we hope our next webinar will be scheduled in June, so please stay tuned on the SOARCE webinar webpage shown here. Thank you again, Emily and Bart. Thank you each for taking the time to join us today, and to all of our participants who tuned in. And we'll end today's webinar.
Thank you, Laura. Thank you, Emily, as well.
Thanks, everyone.